Good morning, everyone. I'm glad to be here to share our recent work, and it's about gen virus detection in the metagenomic sequence. And let me give you a brief introduction about what are the gen viruses. And as their name goes, they are the largest group of viruses we have ever found. And due to their comparable genome size, at the very beginning, we even thought they are bacteria. So, but then we found they cannot replicate without their host, and so they are actually viruses. And why they are important? Because they have a large genome size. They can contain multiple proteins and provide more insights about the revolution or the evolution of the viruses, and also contain some special proteins like the cold adaptation, depth adaptation proteins in the deep marine. And we also found they carry a lot of antibiotic resistant genes. Uh, Although we don't even know whether or why they contain this protein and whether they will express, but they do contain that, and even more than the bacterial phages. So our task is to detect the gen virus in the metagenomic data. So the input will be the sequence, will form the sequencing machine and their DNA sequence. Typically, they will be contig level, and we will feed this contig level inputs into the model, and then the model will decide whether a sequence belongs to the gen virus or other microbes or other viruses. So simply speaking, it will be a binary classification like 1 for the gen virus and 0 for all others. And we always have some challenges when designing a model, right? So in the gen virus detection, the main problem will be the imbalance in data. As you can see, when you try to check the reference genomes, such as in the RedSeq database, we found only around 200 genomes about the gen virus in the database. So it's a very small fraction of all other microbes and all other viruses. And a simple toy example shown here shows that all the data points are in the purple circle. And if you feed all this data to a machine learning model, the model will maybe simply return all the prediction as the purple circle because this will lead to a good performance on an overall data set, right? So there are some simple way to overcome this problem. Like we can random sampling some negative data. For example, here we can only sample the same number of the negative samples as the positive one, as the number of gen virus, and then we can draw a distinction boundary. However, you can see if we use the whole data set, the original data set to draw a boundary, the boundary is typically different from the sampled one, right? So a random sampling usually will lose some important features to generate a more precise decision boundary. And our aims is to find a better decision boundary. For example, we want to find the hard cases that is hard to distinguish by the models. And we also want a better speed, right? Because we have much more negative data. For example, we don't want to align our sequencing data to the whole NR database, right? It's very time consuming. And our idea is borrowed from the Monte Carlo tree search. And I think everyone knows about the AlphaGo, right? The best AI chess player that beats the human player in the chess gaming. So the algorithm behind the AlphaGo is the Monte Carlo tree search. And you can see here is an example how it did the search to win the game. So for each step, it tried to find the optimal options to play the game, and because the future move is depend on the previous move. So it's like a search on the tree. So actually, we have many tree structure in the bioinformatics, and here we use the taxonomy tree. This is because the taxonomy tree automatically group the sequence, group the genomes with their similarity. And intuitively, if we find a hard case, the species has the same taxonomy will also be the hard cases. And if we find the easy case, the species with the same taxonomy or will also be the same, will be the easy case. So here is our framework. It's a reinforcement learning framework. So we have two parts different from other models. Here is the model part. The model will learn how to distinguish the input sequence and draw a decision boundary. And also we have MCTS agent, the Monte Carlo tree search agent. This agent is quite standard in the reinforcement learning. It's not a GPT-like agent. It only takes some computation and iteratively updates the weights in the agent and find the optimal solution. So it's like the model will give the loss, which is the signal or rewards to the agent, and the agent will use it to select the hard cases from the database and then generate new samples for training. And let's go deeper to the Monte Carlo tree search. For the first for the first three steps, we will try to figure out how to find the hard cases from. Sorry. Okay. Okay. So here we're going to find the hard cases on the tree, and you can see there is an interaction between the model and the agent. It will deliver the information, the loss, the rewards to find out which part of the samples, which part of the tree is the hard cases, and update the weight on the tree. And then we do another iteration to find the hard cases. And actually, the algorithm behind the Monte Carlo tree search is this equation. And the QI, you can imagine the QI is the loss of the model, the loss from the model. And maybe a higher loss means harder cases. So it's like we would like to go deeper on the tree and find the sample with our largest loss. And the second term, the right-hand term, this term constrain that because in each iteration we update the loss, right? We sum up the loss, and if we don't consider whether a node has been encountered frequently, we found the agent only select some of the nodes with the largest weight and don't explore the whole tree. So the second term constrain the algorithm to explore wider on the tree. So the first term Q, like we want to go deeper, and the second term we want to go wider on the tree. So the whole space, the whole search space can be explored. And back again, we talk about the agent and oops, sorry. So we talk about the agents, and now we will talk about the model we designed here. And different from the PLM, the protein language model, we actually use a more simplified one. It's a protein cluster-based transformer model. So the first thing is we translate the DNA sequence into the protein, and we map the protein to a predefined protein cluster database. Then we will we will map the protein cluster ID to each of the database. It's like in this format, the word, the token are the protein cluster ID. So it's quite different from the token or the amino acids in the PLM. And this design provides significant several benefits because the PLM usually return an embedding, right? And when you train a model based on this embedding, you will often got only the prediction like 0 or 1. However, that is not quite enough for the users in the microbiome because we want much more information as more as possible. But in the PC-based design, because we align the protein, right, so we can easily to return which protein cluster this sequence has. And the importance of the protein cluster driven by the model and also the protein interactions between the protein clusters. And also we can have a higher confidence results because if you use a PLM, you don't know whether it's aligned to a reference, right? But in this one, we can know the coverage of the proteins on this genome, and then we can output like the confidence is the coverage of the proteins of this core context. And it's very easy to extension because we always want to do some downstream analysis like the final genomic tree. So in our PC design, we can simply return the protein annotations and the user can choose their marker genes to build the final genomic tree. And of course, we did some PLM algorithms before and we released it on the GitHub. We usually get an issue like we don't have GPU cards, how to run it on the CPU. So in the PC design, we can have a faster speed than the PLM and we don't need any GPU cards. And back again, our framework contains two components. The transformer model will provide the loss and the decision boundary for the agent, and the agent will try to find out more harder case to generate a better training set for the model. And finally, when the model is trained, we only provide the user with the model, and that would be very fast to run any sequencing data. Right, and of course, I will show you some benchmark experiments. We benchmark with the, as far as I know, the only two work that can identify the GiantVirus, which one is the viral recall, and it's based on alignment and searching the similarity between the reference genomes. And the second one is a virus sorter. But the virus sorter only provide an extension database for the GiantVirus, so it's not designed for the GiantVirus. And as you can see, we have a faster speed and we have a higher precision. Right, and we also do an ablation study to show how the MCTS works. And as you can see, the MCTS indeed improves the performance compared to the random sampling. We also do some case study, like we sampled the water sediments along the river, and the cities is separated by the three dams along the Yangtze River in China. And we also found many GiantVirus sequences with our tools, and we do some quality check and we do the binning to refine the GiantVirus genomes. And we also find some interesting things in this data. It's like the dam will separate the diversity of the GiantVirus. It's like along the upper string, the diversity increased, but after the first city, Wuhan, in the downstream of the dam, we found the diversity drops significantly. So it might be because the dam will affect the environments. And we also show the ability of GiantHunter, our tool that can generate, that can output the marker genes for the user. So here's the marker gene poly B that is commonly used to create the phylogenomic tree for the Giant virus. And we found many of our newly identified Giant virus are novel. But be careful that because it's based on marker gene, only half of the newly identified Giant virus contain the marker gene. So we still miss some novel one. Yeah, quick summary. We apply the Monte Carlo tree search framework to do the negative sampling. And of course, it's a more general framework that can be applied to other tasks. And we have provided tools that largely improve the accuracy and provide a more user-friendly software. Also, our group provides many tools that solve the problem in the microbiome field. One of it is the web server Phagebox. So the first design of the Phagebox is for phages, but now we extended it to more large field, include all the virus and include tasks like identification, taxonomy classification, lifestyle prediction, and host prediction. And we have a large community, and hopefully it will improve it will advance your research, and we are accept any feedbacks to improve our tools. We also have a poster. It's about assess the quality of the viral sequence. So maybe you have already used the CheckV. And we also benchmark with CheckV here, and to show a higher precision, because we also use the DNA branch to better determine the boundary of the prophages and the chromosome. Yeah, finally, I'd like to acknowledge my professor, Sun Yanni, and also the first author of this work, Fu Chuan. Because of some health problem, I have to present this talk for him. Yeah, and hopefully he will back to our group soon. Yeah, thanks.
Thank you very much for the talk. With time for one question.
Hi, thank you for the interesting talk. So you're using the taxonomy to train on protein cluster annotations, right? Or that's part of the loss function. So are you training for predictions? So is it like a taxonomy profiling tool that's working on some kind of the viral taxonomy? And then how does it tie to MAG connections? If you are imagining that you just get some context that carry those markers?
Actually, in this work, we didn't do in the Giant virus identification detection. We didn't do the taxonomy classification in the model. You mean this one? Sorry.
In this Monte Carlo that we showed, like you're making use of the taxonomy.
I see. So this one, right? So this taxonomy tree is only used for tree search. It's like it's not about the protein cluster. It's only about which sample, which genomes we'd like to choose for training.
I see. So specifically Giant viruses, virus versus
versus all others. Yeah.
Okay. Thanks again.
Thanks.
Move on.